Hello, and I'll be talking now about high definition transcranial direct current stimulation, or HD-tDCS, and describing why this is a very compelling neuromodulation platform because it is low power, it is non-invasive but targeted, and as a result of it, it can be customized and deployed for a broad range of indications. These are my disclosures, and I'll also share the slides and supporting material online. So there are many forms of neuromodulation technologies with different names. These names typically describe how the energy is delivered into the body. So in the case of deep brain stimulation, electrodes are implanted by a neurosurgeon deep in the brain to stimulate deep brain targets. Spinal cord stimulation is another invasive technique that stimulates the spinal cord. Then there are techniques like transcranial magnetic stimulation or electroconvulsive therapy. These are techniques that a patient would need to go into the clinic in order to receive therapy. And then there's this last class of techniques that are low power. And as a result, I want to describe them as wearable in contrast to techniques that require surgery or in hospital treatment. And one of the most researched approaches among wearable neuromodulation techniques is something called transcranial direct current stimulation. And that's what I'm going to be talking about today. Now, transcranial direct current stimulation, TDCS, as the name implies, is a wearable neuromodulation device. Electrodes are placed on the scalp, and a weak direct current, not pulses, and this makes it this unique compared to other neuromodulation approaches. A weak direct current is passed through the electrodes on the scalp, uh, producing current flow through the brain, and this results in the neuromodulation. And it is possibly the most investigated technique device currently in neuromodulation and psychiatry, um, with hundreds of clinical trials published testing this technique on different indications. This is what transcranial direct current stimulation, or TDCS, looks like on me. You can see there are two electrodes, at least always. Positive electrodes are called anodes. Negative electrodes are called cathodes. They're held to the head with a headgear. There's a cable that in this case is running down the side of my head that is connected not to a battery, but to a battery-powered current-controlled stimulator. The amount of energy that's applied is very low. It's typically about 2 milliamps. So 9-volt battery or a little bit more is actually enough voltage to produce this much current across the human head. The individual sessions last for about 20 minutes, and they may be repeated 5 times per week for several times for several weeks, depending on the indication. And you'll also hear the term anodal or cathodal TDCS used. Because there's always an anode and a cathode, those terms actually refer to the fact of whether the brain region that's being targeted is near the anode or near the cathode. Now, regular TDCS uses pad electrodes. These are the electrodes you just saw. They can be 5 by 5 square centimeter pads, and they can be deployed in different positions on the head. For example, in the M1SO montage, which stands for motor to supraorbital. About a decade ago, we proposed that TDCS could, instead of using sponges, could be applied with small electrodes, and we call these high definition electrodes. Now, because the electrodes are smaller, they can be placed closer together, and because they're smaller, you can position more in the head, so you can configure them in different arrays. And one of the configurations we proposed was the 4x1 montage. So this would be the 4x1 high definition TDCS montage. And we developed these montages using computational models. So the images you're seeing here are actually MRI-derived models of the human head. Specifically, this is of my head, and you can see both conventional TDCS and high-definition TDCS. And these models predicted that when regular TDCS is applied, the current flows through these two pads, impacting much of the brain. So current does get into the brain, but with conventional TDCS, the stimulation is not focal. But with high definition TDCS, the stimulation is focal. And in fact, whereas with regular TDCS, the entire area between the two pads is washed in current, with high definition TDCS, the current is restricted to the perimeter of electrodes that make the outside of the 4x1 ring. In fact, the smaller that outer ring, the more focal the stimulation. So this technique was initially developed and proposed through computational models. It was then developed into an instrument and has since then been widely tested for a range of indications, just like TDCS is. So with regular TDCS, because of the diffuse current flow pattern, we're really thinking about circuit therapeutics. We're thinking about current that polarizes many nodes in the brain, producing a neuromodulation effect. While with high definition, it's actually possible to be non-invasive and targeted at the same time. Now, I've shown you these models. I should say these models have been extensively validated using a variety of different approaches. In this case, we used intracranial electrodes in human subjects. These were subjects who had epilepsy and were candidates for resection. So they had the electrodes implanted into these arrays implanted into their brain for the purpose of pre-surgical mapping. But we used these same electrodes to map the flow of current flow during transcranial direct current stimulation. And as summarized in detail in this paper here, the short of it is that the models match the recordings. So we believe these models, these images that I've been showing you, are reliable. In fact, there's been additional validation techniques that have been done. So as I started the presentation, I think that high definition TDCS has several unique features as a neuromodulation tool that make it very appealable for deployability. The first one is that it's considered very safe and highly tolerated. It's considered safe enough that it's applied routinely in healthy individuals, healthy college students. So it is non-significant risk, minimal risk, and it's very tolerated. The main adverse events are itching, tingling that occur during stimulation, and these resolve right afterward. Like regular TDCS, high definition TDCS is low power, therefore it can be provided with a small battery powered device. And it combines both non-invasiveness and being targeted. And I want to talk more about how this targeting is achieved. There's actually 4 different types of targeting that could be considered in the context of high definition TDCS. And I'll briefly describe each one of them. One is more anatomical based targeting, one is image based, and the last 2 actually get at the mechanisms of TDCS and how they can be leveraged to enhance targeting even further. So starting with this first idea of anatomical targeting, quite a few years ago, we developed algorithms that allow optimization of high definition TDCS to reach any brain region that can be simply selected. And this is the publication that established that algorithm. And as I just mentioned, a brain region is selected, a region of interest, and then the software prescribes how many electrodes, where those electrodes should be, and how much current should be in each electrode to optimally stimulate that region, be it superficial or be it deep. On top of that, because different people's heads are different, and there could be situations where a subject may have a skull defect or a brain injury, the software, by uploading individual MRIs, can do this optimization on a subject-specific level. And then focusing on TDCS, the same algorithms, the exact same math can equally be used to optimize alternating current stimulation and various forms of multi-current stimulation, as well as multi-target stimulation. So this is the anatomical targeting. Now, there's another approach that has been researched by for many years and was addressed in my lab as well, which is the use of EEG to inform stimulation. And this is actually a very old idea called reciprocity. And the basic idea is that the brain is somehow made active. Let's say the individual is doing a task. As the brain is active, that activity is detectable on the surface of the head using electroencephalography, EEG. So this is the electrical activity generated by the brain picked up on the scalp. And a long-standing idea was that you could somehow detect this brain activity at the scalp and then invert the electrical signal that you were recording into an electrical stimuli that you were applying in order to stimulate the same brain regions that were generating the activity. So if the task engages a specific region of interest, and then that activity of that region of interest is manifest on the surface of the scalp with a particular pattern, we can take that pattern, invert it, reapply stimulation to retarget that same region. And though this idea has been along for a long time, it was only through the incorporation of our individualized MRI models that we were able to develop a closed form solution for this problem that's explained in the reference that's cited here, where in a blinded way, but provided the individual MRI, you could record an EEG in a receiver and then apply the stimulation directly. And so now we can say, well, you can pick a brain region or don't. You can simply record brain activity and optimize stimulation based on the brain recording as well. So both, and the latter one does also support closed loop stimulation. So moving ahead quickly, I just quickly described two forms of doing what you could call anatomical targeting, in the sense that you are focusing on a specific region. Now, how you find that region can be based either on simply selecting that region or using an imaging technique like EEG. But you can go still deeper. And one emerging idea in transcranial stimulation that requires leveraging high definition stimulation is sub-gyri targeting. And this idea gets to the fact that as current flows through the brain, it doesn't just have an intensity at a given region, it has an orientation. And several investigators, including the citation showed here, have shown that depending on how the current flows through a brain region, so the direction that it flows in, you can produce very different neuromodulation outcomes. So the same brain region exposed to current flowing one way versus flowing the other way will be influenced in a different way, in a way that ultimately provides tremendous nuance in how current can be guided to produce specific outcomes. And this is one area that I think is very exciting, and I call it sub-gyri targeting, because it's not just focusing the current to a specific gyri, but actually thinking about how structures within that gyri can be independently modulated in a way that I think is very unique to high definition TDCS. So that's sub-gyri targeting. The final layer is something that we call functional targeting. And let me introduce that in a few minutes. So we're now going to go from anatomical targeting, which really has focused a lot on identifying a region of interest, to something called functional targeting. So traditional or other forms of neuromodulation, I could say, like high intensity transcranial electrical stimulation, transcranial magnetic stimulation, electroconvulsive therapy, and also all forms of invasive stimulation, they're very high intensity and they apply pulses. So a pulse is applied into a region of interest, a bunch of neurons are exposed to that pulse, and they fire an action potential. And these pulses tend to be repeated. So if the pulses are being applied at 1 hertz or once per second, the neurons fire 1 time per second. If the pulses are applied at 100 hertz or 100 times per second, the neurons fire at 100 times per second. So almost all forms of neuromodulation other than TDCS and also TACS, another form of low intensity transcranial electrical stimulation, but all other forms of neurostimulation are essentially blasting little pockets of tissue and making them fire. So this is referred to as super threshold stimulation. You are above thresholds for these neurons, you are making them fire. And any neuromodulation you see, any therapeutic outcome of all of these techniques, all these super threshold non-invasive techniques, but also every invasive technique, is a consequence of this overdrive, of this super threshold stimulation. Now with subthreshold stimulation, among which really we're focusing on transcranial direct current stimulation, but there are other variants as well, the current is actually very low. It's about 2 milliamps. And when this current reaches a brain region, the intensity of current that surrounds neurons, when they're quiescent, is not enough to make them fire. So in the case of, let's say, transcranial magnetic stimulation, the intensity or the electric field that is generated around neurons can be over 100 volt per meter. But with TDCS, and other low-intensity transcranial techniques, the intensity of current or the electric field generated around neurons is less than a volt per meter. And so when these neurons are not firing, are not already active, they're not made to fire because the stimulation is much too low intense. So there is no de novo firing, making neurons that are not already firing fire. However, what I'm going to show you is that we know that the brain is never inactive. The brain is always active. And in fact, we can drive the brain to certain patterns of activity, for example, by training people or making someone practice something. So the brain is already in an active state. And it turns out that when you apply low intensity electrical stimulation to an active brain, those neurons that are already active sense that electricity and can be modulated. So the current is actually going to a wide region of the brain. Inside that brain region, there are neurons that are both active because you're doing a task, and neurons that are not active because they're not involved in that task. Current spans around all these neurons, but only the neurons that are already active sense the current. Only the neurons that are already active are modulated by the current. And so what you have now is you have a situation where you are preferentially stimulating, preferentially modulating only active neurons while sparing inactive neurons. And so again, going to the example of a task, if an individual is engaged in a task, if they're practicing something, and that practice is engaging specific neurons in the brain that could be in different parts of the brain, we now apply weak transcranial direct current stimulation. We apply current through maybe much of the brain, but only those neurons in different parts of the brain that are involved in the task are in a state where they can now be sensitive to the stimulation. Now, this is a cartoon idea. In my lab, we've actually been unpacking this using animal models at the cellular level for decades. Briefly, this is what the animal model looks like. This is a brain slice preparation, an acute brain slice preparation from a rodent that is maintained in a dish. The two wires you're seeing at the top and the bottom are the stimulation wires, and we can use a variety of microelectrode and microprobing techniques to assess how weak direct current affects these systems. We can apply current in different directions depending on how we connect the positive and negative terminal to these wires, and so we can have a direction that is analogous to the nodal direct current stimulation, or we can have a direction that's analogous to the cathodal direct current stimulation. Here's one experiment that is very powerful and illustrative. There's two components of this experiment. The first part of the experiment is unrelated to direct current stimulation. It is the induction of long-term potentiation with theta burst stimulation. This is what's happening here. We have a system, and that system has two populations of neurons that are connected to one another. The strength of that synaptic connection is measured by this excitatory postsynaptic potential, or EPSP. So the amplitude of the EPSP is an indication of how strongly this region is synaptically connected to this region. Now, if you go into that system and you briefly but intensely blast it with theta burst stimulation, this is not direct current stimulation, this is very intense microshocks, what you can find is that after this microshock wave is over, the connectivity between these two brain regions has increased, which is measured as an increase in the strength of the excitatory postsynaptic potential. So this is classic long-term potentiation, theta burst pattern stimulation produced long-term potentiation that shows that the strength of connectivity between two brain regions is plastic, and when you have a lot of activity, you can make it stronger. Now, we combine the induction of long-term potentiation by theta burst stimulation with weak cathodal or nodal direct current stimulation. This is the result of that experiment. So on the left, you have a cartoon schematic of the experiment. You can see there is a microelectrode that is being used to tetanize with theta burst the system. And we can either apply the theta burst stimulation on its own. We can apply it at the same time as a nodal direct current stimulation, or at the same time as cathodal direct current stimulation. And the black lines in the middle tray show you that initially, prior to tetanization, which happens at t=20 minutes, we have a stable EPSP amplitude this case, it's a field EPSP. It's measured extracellularly, but it is stable. And we're actually normalizing these experiments, that stable baseline of 1. And then at t is equal to 20 minutes, this tetanization is applied. And plasticity is produced. And the black dots, which you can see in the background, represent the theta burst on its own. You see an increase of about 70%. And for as long as we run this experiment, so over an hour later, some level of plasticity connectivity boost persists. If the theta burst stimulation is applied at the same time as the anodal variant of TDCS, it is more effective. You produce more plasticity, a more robust increase in connectivity. And you can see that in the pink trace. So the pink dots are above the black dots. What that means is the same tetanization, the same theta burst, and that gives you more plasticity, so more bang for your buck. The cathodal version of TDCS, and the blue does the opposite. It makes the theta burst stimulation less effective. But a very important idea here is that if we apply the direct current stimulation on its own, without theta burst stimulation, no plasticity is produced. What that means is if you have one population that is inactive, it is not undergoing plasticity, and another population that is undergoing plasticity, and you apply direct current stimulation to the whole system, this system here that is not undergoing plasticity will not be influenced, and this system that is undergoing plasticity will experience boost, will experience the benefit. Only those synapses that are already undergoing plasticity, and plasticity is considered an analog for learning, only those synapses that are already undergoing plasticity receive the benefit of direct current stimulation. This is functional targeting, and we also describe this as a boost of Hebbian plasticity. And the relationship between direct current stimulation and the enhancement of Hebbian plasticity has been unpacked with tremendous detail, and the reference cited is just one source. And again, this paper shows in tremendous detail why direct current stimulation boosting of LTP follows Hebbian rules such as associativity. And again, the biggest point is that this is functional targeting. Only systems, only networks that are already active because the person is doing something, because they're undergoing rehabilitation, because they're undergoing training, only those networks get the benefit of TDCS. And again, this is the point that there is associativity at the cellular level, and this is important in the sense that it would support the notion that of generalization during training and TDCS. So I just, in summary, I showed that high definition TDCS is a compelling neuromodulation tool. It has features that are really unique in the neuromodulation space. First of all, it is safe and highly tolerated. It is wearable. Most people won't even feel that it's on. It's battery powered. As a result of that, it's portable as a result of that. It's very easy to integrate it with other wearable techniques as well. It is non-invasive, it is transcranial, but it is also targeted. And that targeting takes on a whole new level when you start realizing that direct current stimulation, because it is low intensity, can be shaped and manipulated in a way to leverage sub-gyri targeting and also functional targeting. So that was my presentation, and thank you.